Welcome to Inert Chemistry. In today's video lecture, we will discuss the transamination reactions of amino acids and their importance. In the previous video, we started the metabolism of amino acids and we studied that the amino acid metabolism is divided into two parts: the metabolism of amino group and the metabolism of carbon skeleton. During the metabolism of amino group, the first step is transamination, during which the transfer of amino group from an amino acid to keto acid occurs. In today's video, we will discuss the transamination in detail. Transamination. Transamination is defined as transfer of amino group from amino acid to keto acid. For example, here a general amino acid is shown and it contains NH3 amino group. It is a donor atom and it donates the amino group to a keto acid. Alpha ketoglutarate is a keto acid. The amino group from an amino acid is transferred to keto acid. As a result of this transfer, a new amino acid is formed, that is glutamate, and a second keto acid is formed. This process of transamination is a reversible process. It requires transaminases. Transaminases are actually enzymes that catalyze the transamination and coenzyme pyridoxal phosphate. This PLP is actually derivative of vitamin B6. There are two important transamination reactions. The first one is catalyzed by the aspartate transaminase or AST. In this reaction, the amino group is transferred from L-aspartate to alpha ketoglutarate, leading to the formation of L-glutamate and oxaloacetate. This reaction is catalyzed by the transaminase that is called aspartate transaminase or AST. This aspartate transaminase or AST, it was previously known as SGOT. SGOT stands for serum glutamate oxaloacetate transaminase. This reaction can also be written as L-aspartate plus alpha ketoglutarate forms oxaloacetate plus L-glutamate. The AST or SGOT is the enzyme that involved in the catalysis of this reaction. This AST or SGOT is having clinical significance because it involves in the diagnosis of heart diseases. In case of heart disease, the level of AST or SGOT alleviates. The second important transamination reaction is catalyzed by alanine transaminase or ALT. In this reaction, amino acid is transferred from L-alanine to alpha ketoglutarate, leading to the formation of L-glutamate and pyruvate. This reaction is catalyzed by alanine transaminase or ALT. This ALT was previously known as SGPT. SGPT stands for serum glutamate pyruvate transaminase. The reaction can also be written as L-alanine plus alpha ketoglutarate forms pyruvate plus L-glutamate. The ALT or SGPT is having clinical significance because it is used for the diagnosis of liver diseases. Now we discuss the various features of transamination. This reaction was discovered by Brostin and Britzman in 1974. These reactions are reversible reactions. These reactions are mostly occur in liver, kidney, heart and brain. They required coenzyme pyridoxal phosphate. During this reaction, there is only transfer of amino group from amino acid to keto acid. In this step, there is no formation of ammonia because ammonia is formed in the next step, that is deamination. The amino groups are eventually concentrated in glutamate. Glutamate acts as a storehouse of amino group from various amino acids. Glutamate is the only amino acid that undergo oxidative deamination and release free ammonium ion. There is the production of non-essential amino acids. The major importance or significance of transamination is that it leads to the formation of other non-essential amino acids. The specific transaminases that are used in this reaction are ALT, GPT, AST or GOT. There are some amino acids that do not participate in transamination. Must remember, these includes lysine, threonine, proline and hydroxyproline. The transamination reactions are not restricted to only alpha amino group because the delta amino group of ornithine forms glutamate gamma semialdehyde. So we can say that this transamination reaction is not restricted to only alpha amino group, but the delta amino group can also undergo transamination reaction. Now we discuss the various biological significance of transamination reaction. This transamination reaction is having various biological significance. For example, it is the first step of catabolism. During the catabolism or degradation of amino acids, first of all, the amino group from amino acids is removed and the remaining carbon skeleton then undergo different catabolic pathways. And by removing amino group from the amino acid, ammonia is formed, which leads to the formation of urea through urea cycle, and then this urea is excreted from the body. The second important biological significance is the synthesis of non-essential amino acids. All the non-essential amino acids can be synthesized by the body by the help of transamination reactions. The third important significance is the interconversion of amino acids. If amino acid number 1 is high and number 2 is low, the amino group from amino acid number 1 is transferred to a keto acid to give amino acid number 2, so that both the amino acid can be equalized. There is also some clinical significance of transamination. AST and ALT. These are the important markers of liver injury. Here are some exceptions. There are some amino acids that do not undergo transamination. These include lysine, threonine, and proline. They do not transaminate. They follow the direct degradative pathways. Thanks for watching this video.